Hi, my name is Petr Smirnov, and today I'd like to tell you about my poster on the meta-analysis of preclinical pharmacogenomic studies for biomarker discovery. Now, this work is really motivated by the fact that in many precision oncology trials to date, clinicians still struggle to match patients to treatments based on the genomic profiling of the patient's tumor. And we believe that this is due to the fact that we've still only barely scratched the surface of understanding how the molecular, genomic, and transcriptomic features of a tumor affect drug response. And this is evidenced in the fact that for the majority of anti-cancer drugs on the market, we don't have a matched genomic biomarker to identify patients that will respond to it. We seek to address this issue by looking at preclinical pharmacogenomic screens, where cancer cell line models have undergone both treatment with a large panel of anti-cancer compounds to measure their sensitivity to treatment with each compound, as well as comprehensive molecular profiling. And we ask the question which of these molecular features are most predictive of response to any particular drug, and as such, try to discover biomarkers of drug response. We then also try to build evidence that the biomarkers that we discover could be applicable in the clinic by looking either at more complicated cancer models, such as patient-derived xenografts, or retrospective clinical data. This project in particular builds upon efforts in the BHK lab in developing the pharmacodb.ca database and web app. For this database and web app, we went out and collected 10 of the largest published pharmacogenomic studies released to date. We've downloaded the raw data, reprocessed with standardized pipelines, made sure all the identifiers used in the studies agree cross studies, and we make this data available for researchers to use in their own research. Given that we've collected all this data and made it as comparable as possible, we now want to do meta-analysis across studies. And for this project, we focused on finding single gene expression markers of drug response across 7 studies where we have independent drug response and gene expression profiling. And we are able to look at millions of individual gene to drug associations. When we apply the pipeline that we've designed, we first look at whether any of these associations are significant in each particular data set, for which we find just over 7,800 significant associations. And then we apply meta-analysis to see whether these associations are supported by the consensus of all 7 data sets. And here we find our first interesting result. Almost half the associations that were discovered in any particular data set are not going to validate after meta-analysis, suggesting that looking across studies and integrating data from multiple sources is actually helping us pick out the robust and reproducible associations. We then, from this list of 4,338 putative biomarkers, are trying to understand what the biology behind why some of them may predict drug response is, as well as to build evidence that some of them might be clinically relevant. And for the purposes of this short presentation, I'm going to focus on a case study about the retrospective validation. This particular case study is about the expression of the ODC1 gene among breast cancer cell lines and its association with paclitaxel response. For predicting paclitaxel response in cancer cell lines, we actually found 3 genes whose expression correlated with sensitivity. Thankfully, we were able to collaborate with Dr. Dave Cheskon's lab and use an experiment which they ran where they took patient-derived xenograft models and treated them with paclitaxel to see which of these xenografts are going to be most sensitive to Taxol treatment. And we're able to show that ODC1 is the one biomarker that significantly predicts response both across cell lines and patient-derived xenografts in this case. Given that this biomarker validated in the patient-derived xenograft data, we then went to see whether we could find any retrospective clinical data to help build further evidence. And we were able to leverage the MAQC2 cohort, which was a cohort of 278 preoperative breast cancer patients, which underwent neoadjuvant treatment with chemotherapy, which included paclitaxel. And among these patients, high expression of ODC1 was also predictive of good response to the neoadjuvant therapy, giving us an AU-ROC of 0.73. As such, this is one of those examples where we were able to combine discovery in cancer cell lines, where we have a lot of power and can look at many biomarkers, with a relatively smaller 50 patient PDX data set and smaller clinical data set to discover a biomarker that in cell lines and show that it can validate in clinical data. So in summary, we've implemented a pipeline for the meta-analysis of preclinical pharmacogenomic studies. We've seen that nearly half the discoveries supported by any single study are not going to be significant for meta-analysis. And we've shown how you can do discovery in cancer cell lines and then use smaller and more targeted data sets to build evidence for the translation of these discovered associations back into the clinic. Finally, I should acknowledge the members of both my labs that have helped me throughout this project, our collaborators, and our funding sources. And I hope that you'll come check out my full poster sometime during the conference.